CRISPR and gene editing. Rewriting life, the discovery of CRISPR-Cas9 technology has revolutionized the field of genetics, offering scientists a powerful tool to edit DNA with unprecedented precision and efficiency. From curing genetic diseases to engineering crops and potentially reshaping entire ecosystems, CRISPR has opened the door to possibilities once confined to science fiction. However, with great power comes great responsibility, as ethical, legal, and safety concerns emerge alongside its revolutionary potential. What is CRISPR-Cas9? CRISPR, short for clustered regularly interspaced short palindromic repeats, is a natural defense mechanism found in bacteria. Bacteria use CRISPR to identify and cut the DNA of invading viruses, providing protection from infection. Scientists have harnessed this mechanism, particularly with the Cas9 protein, to create a gene-editing tool capable of targeting and altering specific DNA sequences in plants, animals, and humans. The process involves designing a guide RNA, gRNA, that matches the target DNA sequence. Cas9, an enzyme, is then directed by the gRNA to the desired location in the genome where it makes a precise cut. Once the DNA is cut, natural repair mechanisms can be exploited to either disable a gene, correct a mutation, or insert new genetic material. Applications of CRISPR in medicine. One of the most promising applications of CRISPR is in the treatment of genetic disorders. Diseases caused by single-gene mutations, such as sickle cell anemia, cystic fibrosis, and Huntington's disease, are prime targets for gene editing. Sickle cell anemia. Clinical trials are underway to use CRISPR to correct the defective gene responsible for producing abnormal hemoglobin. Early results have shown promising outcomes, with patients experiencing reduced symptoms. Cancer therapy. CRISPR is being used to engineer immune cells, such as T cells, to better recognize and attack cancerous cells. This approach is being tested in immunotherapy treatments for leukemia and solid tumors. Gene therapy for rare diseases. Beyond common genetic disorders, CRISPR holds potential for treating rare conditions like Duchenne muscular dystrophy by repairing mutations in the dystrophin gene. Agricultural advancements with CRISPR is transforming agriculture by enabling scientists to develop crops that are more resistant to diseases, drought, and pests. Traditional breeding methods can take years to achieve desired traits, but CRISPR accelerates this process by directly modifying the genes responsible for specific characteristics. Disease-resistant crops. Scientists have edited the genomes of rice, wheat, and bananas to make them more resistant to common plant pathogens. Enhanced nutrition. CRISPR has been used to boost nutrient content in crops such as tomatoes and rice, potentially addressing malnutrition in regions where access to diverse diets is limited. Climate-resilient crops. By editing genes related to water usage and heat tolerance, CRISPR can help farmers grow crops that are more resilient to climate change. Environmental applications. CRISPR is also being explored as a tool to address ecological challenges. One of the most debated applications involves gene drives, a technique that ensures a specific gene is passed on to nearly all offspring, potentially altering or eliminating entire populations of species. Controlling invasive species. Gene drives could be used to suppress populations of invasive species that threaten biodiversity, such as mosquitoes carrying malaria or invasive rodents on islands. Conservation efforts. Scientists are investigating whether CRISPR can help revive endangered species or improve genetic diversity within shrinking populations. However, environmental applications raise concerns about unintended ecological consequences. Altering ecosystems could have cascading effects, making it crucial to conduct thorough risk assessments before deployment. Ethical and social considerations. CRISPR's ability to rewrite the genetic code poses significant ethical dilemmas, particularly when applied to humans. The possibility of editing human embryos has sparked global debate about the boundaries of genetic engineering. Germline editing. Editing the genes of embryos could prevent hereditary diseases, but it also raises concerns about unintended mutations and the potential for designer babies, with selected traits such as intelligence, appearance, or athleticism. Access and equity. As with any medical breakthrough, there is a risk that CRISPR-based treatments could exacerbate social inequalities if they are accessible only to the wealthy. Consent and regulation. Questions arise about who should decide how CRISPR is used, especially when it comes to editing the genes of future generations. International guidelines and regulatory frameworks are being developed to address these concerns. Safety and technical challenges. Despite its precision, CRISPR is not without risks. Off-target effects, where unintended parts of the genome are edited, could lead to harmful mutations. Scientists are actively working on improving the accuracy and specificity of the technology to minimize these risks. Off-target mutations. New variants of CRISPR, such as base editors and prime editing, are being developed to increase precision and reduce the likelihood of unintended changes. Immune reactions. Some studies have shown that the human immune system can recognize and attack the Cas9 protein, potentially limiting the effectiveness of CRISPR-based treatments. Researchers are exploring ways to overcome this issue through the use of alternative proteins. The future of CRISPRase. CRISPR technology continues to evolve. Its potential applications seem limitless. Scientists are exploring possibilities, such as editing microbial genomes to combat antibiotic resistance, engineering tissues for organ transplants, and even editing the microbiome to treat gut-related diseases. However, responsible development is key to ensuring that CRISPR is used safely and ethically. Public engagement, transparent decision-making, and international collaboration will be essential to navigating the challenges posed by this powerful technology. Conclusion: CRISPR-Cas9 has undoubtedly revolutionized the field of genetics, offering unprecedented opportunities to improve human health, food security, and environmental sustainability. Yet the technology's immense power requires careful oversight to balance innovation with responsibility. By addressing ethical concerns, ensuring equitable access, and minimizing risks, society can harness the full potential of CRISPR while safeguarding the future of life on Earth.